This video is going to get you through the first half of chapter 28, which I have called the baby making chapter, because it's essentially about how do we make babies. You got to remember all of the anatomy from the female reproductive system, because a lot of that stuff is going to come back in during this chapter, so just keep that in mind as we start going through it. First off, you just have a few definitions to get down, but I wanted to show you this GIF. A lot of what we're going to talk about you can see happening as this cycles through. So what happens is when a sperm fertilizes an egg, you get a zygote. A zygote is a single fertilized cell. In a human being, it sticks around for about 24 hours, and then after that, it starts a process known as cleavage. And that structure starts to have that one single cell divide by mitosis. So one cell becomes two cells becomes four cells becomes 8 cells, and so on. Eventually, we make a structure that is a solid ball of cells. Now, it is about the same size as the original zygote, it's just made out of more cells now. When it's a solid ball of cells, like right here, it's called a morula. We continue to make more cells, and the cells start to migrate out to the outer portions, and so we end up with like a hollow ball. That hollow ball is called a blastula, and you can see that right about here in the H section. There's numerous names to parts that happen in the blastula. The outer layer of cells is called the trophoblast. That inner little cluster of cells is called the inner cell mass. We'll talk about all of those as we get going. Now, all of the cells at this point, they are all exactly the same. When they talk about embryonic stem cells, these are the cells they're talking about. Any of these cells can become anything they want to be. It can become a nervous system cell. It can become an epithelial cell, whatever. What happens next, though, after we formed a blastula is some of the cells start the process of differentiation, and they start to choose a path, and they go on that path during a process known as gastrulation. Gastrulation ends up forming just 4, and epiblast is not something that you guys have to worry about, but we form 3, inner germ layers. We've got the ectoderm, the mesoderm, and the endoderm, and we defined all of those in AMP1, but just a reminder, the ectoderm is what forms epithelial tissue like your epidermis. It also forms your nervous system. Mesoderm is what forms connective tissue and muscle, and then the endoderm is what forms your mucous membrane. So all of these tissues develop during the process of gastrulation. After we have those tissues, the cells are going to further specialize, and then we're going to actually start making organs eventually. You can see that starting to form as we're sitting right up in this region right here. So the embryo folds up, we get a neural tube. The neural tube ends up becoming your nervous system, your brain, your spinal cord. If that doesn't seal up correctly, we talked about spina bifida a little bit last semester. Next up, usually, let's see, I'll come back over here. Right about in this stage is when the baby is going to implant in the uterus. And then this whole time that we're doing this, we're also making the placenta, we're making the umbilical cord, we're getting attached and everything is all nice and happy. Umbilical cord really starts to form well as you get up here by V and W. Okay. As we get into this true embryonic stage, you're starting to see structures that are starting to look like actual appendages. Like you can see arm buds and leg buds. You can tell there's definitely a head side and a tail side. And yes, human beings do have a tail embryonically. We also have gills, which is a nice weird thing to have. So we've got those going on for a little bit too. After about 9 weeks, though, you can tell this is a human baby at this point, so the name changes right here to become a fetus. Most of our organs are already in place, and so from that point on, we really just start to refine things and make them all get bigger until the point where we actually give birth. And so all of this chapter is a summary of all of the stuff that's happening in that GIF as it cycles through. All right. I'm going to try to link this video. It's a salamander going through all of those processes, and so instead of playing it here, I'll just try to post a link to it so you can see it. Okay. Pregnancy is just the period in time when a woman does have a child implanted into the uterus. The gestation period for human beings, a healthy gestation period is considered to be between 38 and 42 weeks. If you give birth before 38 weeks, it's considered a premature baby. If you last through 42nd week, then they're going to do inducement so that you can go ahead and give birth. Women usually start complaining that things are very uncomfortable closer to 32 weeks, and so women aren't going to let it go after 42. We've been miserable for like a month and a half at that point, so we just want the baby out of us. Embryo. So embryo is from conception until the point where we're about 8 weeks old. Now, what I like to kind of say is the embryo is the period in time when the baby could be anything. The fetus, you can tell it's a human being. For example, the human embryo looks a lot like this, yet this is a chicken embryo, and most people wouldn't be able to tell the difference between a human embryo and a chicken embryo because they look very, very similar to each other. But again, fetus, you can definitely tell there's babies going on in there, and they're baby humans. This does happen to be twins, and one amniotic sac. That's called monoamniotic when they share one sac. Alright, after that you have how long is an oocyte viable? About 24 hours. How long is sperm viable? Somewhere between 12 to 72 hours. It kind of depends on where the sperm get released. Like if they get released outside of a body into, let's say, a dirty sock, then it's not going to last as long. But if they do get released in the female's reproductive system, they can last a little bit longer. This is just me saying once the travel restrictions are allowed again, or lifted, and you're allowed to travel again, there is a museum in one of the Scandinavian countries. I think it's Finland, but I really don't remember for sure. But it's a phallic museum. It's a museum of nothing but penises. And this is the penis of a sperm whale, just in case you've ever wanted to see what one looks like. Alright, where should fertilization occur? At the distal end of the fallopian tubes. We kind of mentioned that in the last chapter. This is just us reminding you again. If it doesn't happen there, we run that risk of ectopic pregnancy that we had talked about in the last chapter. Fertilization is when the sperm actually binds to a receptor on the egg and then releases the nucleus into the egg, and then a zygote is the fertilized egg. And again, a zygote doesn't last very long for human beings. It's only about 24 hours. Next up, you have why does it take millions of sperm to fertilize an egg? So, technically speaking, it does really just take one sperm to get to an egg, except that there are a couple of different problems that happen. So when I say technically, I mean a doctor can inject one sperm into one egg and you can get a baby out of that. But from a natural standpoint, whenever a man ejaculates into a woman, first off, the woman's immune system hates all of that, and the vagina is very acidic, and sperm hates acidity, and so those two factors are going to kill a lot of sperm. Second, sperm are very metabolically active. They require a lot of energy, and they run out of it fairly quickly, and so they run out of energy before they get where they're going. There's a layer of cells that gets ovulated with an egg, some of those granulosa cells that we had talked about earlier. We have to eat through those before a sperm can actually bind to a receptor, and it takes hundreds of sperm to actually open a receptor up on the egg, and so that's a factor that's going to play in there too. This is why one of the things that can lead to infertility in a couple is a guy have a low sperm count. If you don't have enough, you're not going to be able to break down enough of the granulosa cells around the egg, which means a sperm will never actually be able to bind to a receptor. There used to be a game, I haven't looked for it lately, where you could pretend you're the woman's immune system trying to shoot sperm, trying to get to an egg, and so y'all can look for that if you're bored during this quarantine. Okay, there's some functional changes that need to happen to a sperm after ejaculation. One, they need to get much better at swimming really fast. So while they practice that a little bit in the guy, now it's competitive swimming and there's sharks chasing them, the sharks being the woman's immune system, so they have to get much better, much faster. Second, at the head end of the sperm, there's a little bag of enzymes. It's called the acrosome. So I mentioned those granulosa cells a second ago. We have to essentially eat those cells, and the acrosome contains the enzymes for sperm to eat those cells. And so we normally keep them inside of that bag, the acrosome. So part of capacitation is weakening that bag so that the sperm's going to be able to kill the granulosa cells once it does reach the egg. So it swim faster and start to weaken the acrosomal membrane, so we'll be able to release those enzymes. Next up, I gave you this series of pictures. I know it doesn't show up super well. I don't know why I chose this picture, but it's the blue words that are on the picture that you're having to label on the series that you've got coming up. I gave you these pictures from the book to try to help you understand, again, why does it take millions of sperm and what actually happens when a sperm reaches an egg. So here we've got hundreds of sperm. We've got an ovulated egg with the granulosa cells around the outside surface that are going to have to be consumed so that a sperm will be able to bind to a receptor. So these little Y-shaped things, these are the receptors, and they are hidden behind several different layers. So we've got layer upon layer of granulosa cells, and some books call this the jelly coat. Your book calls it the zona pellucida. It's kind of this slime layer on the outside surface of the egg that's covering these receptors, and the sperm have to digest through that as well. Once a receptor gets freed and it's available, a sperm can bind to that and then one sperm will fertilize that egg. But we have to get through all of those barriers first. And so that's what's happening in the first set of pictures. So we've created a pocket in the granulosa cells in this picture. So you're seeing acrosomal reaction. This one sperm is releasing enzymes to digest through the zona pellucida or the jelly coat, whichever one you want to call that. Next, one sperm binds to one receptor, and that triggers a little depolarization event, which we're going to talk back about here in just a little bit. But once that sperm binds to a receptor, its membrane fuses with the egg's membrane, and then the nucleus is allowed to enter the egg cell so that it can go find the egg's nucleus, and then they can bond together to make the actual zygote nucleus. Now, because it is critically important that one sperm and only one sperm is allowed to enter into this egg, there's a fast block to polyspermy, and then there is a slow block to polyspermy. The fast block is a depolarization event. Remember, we talked about that back in muscles and the nervous system last semester. It's that little electrical charge that happens so that a nerve can send an action potential down the nerve. Same thing happens here, and that little electrical jolt essentially sort of shocks the other sperm away, and they can't bind it because of that little shock. Now, that jolt is short-lived, so the other thing that that jolt does is send a message down the plasma membrane of the egg to destroy all receptors, because we've already got a sperm that enters. We don't want any of the other receptors to allow another sperm to get in, and so all of the receptors all the way around the egg get destroyed as well. So all of those are the blocks that prevent multiple sperm from getting into an egg. So I kind of just described this stuff, but why is it so important that only one gets in? You can think of it as a simple math problem. Your cells are supposed to have 46 chromosomes. One egg has 23, one sperm has 23. Well, what would happen if two sperm got in? We have 60-something chromosomes inside of that cell, 69. But we don't want that to happen, because a baby with an abnormal number of chromosomes is usually not going to form, and it will just get lost with the menstrual flow. If it does start to form, it usually ends up resulting in a miscarriage fairly early on. Babies just cannot survive with a whole extra set of chromosomes, and so only one sperm is supposed to enter an egg. What is the final trigger for the egg to complete meiosis? It's entry of the sperm. Okay, so I kind of mentioned this stuff as I was going over that GIF at the very beginning. One of the things that I want you to notice is in this picture, the zygote, the 8-cell stage, the morula, the blastula, they are all the exact same size, but the cells that make up the structure get progressively smaller and smaller and smaller and smaller. That's what is meant by cleavage. Cleavage means like you take a meat cleaver and you divide it in half over and over again. So a zygote divides by mitosis to make a 2-cell stage. The 2-cell stage divides to make a 4-cell stage. The 4-cell stage divides to make an 8-cell stage. And we just keep doing that. Cleavage is the process that gets us from a zygote to a blastula to eventually a gastrula a little bit later on. So morula is just a solid ball of cells. If you really felt like you wanted to count the number of cells to figure out what you were at, it's somewhere between 16 to 64 cells. Blastula is a hollow ball of cells. It's more than 64 cells, and I don't know if anybody's counted how many there can be, because there can get to be hundreds, if not thousands, of cells in a blastula. The other thing that I'm telling you about the blastula is this is the structure that's going to implant into the uterus. A zygote can't do it, and a gastrula can't do it either. We've got to be at the blastula stage in order to implant into the endometrium and actually get mom pregnant. Okay, looking at the anatomy of a blastula. We've got an outer ring of cells. These are called the trophoblast. The trophoblast is going to end up becoming something called the chorion. The chorion is going to end up making the baby's portion of the placenta. And so by the associative property, that means the trophoblast helps make the baby's portion of the placenta. Next, we got this little blob of cells on the inside. That's the inner cell mass. This is what's going to actually become the baby. And it doesn't look like very much, but it will later on look like a lot more. So said this earlier, it's going to become the chorion, which is going to become the baby's portion of the placenta. Becomes the baby. I feel like I just said all of that, but there you go. It makes the baby. Okay, and then after that, I gave you this picture to go ahead and label. First off, we've got where should fertilization occur. Once again, that's happening way out off over here, and it's something that you have to label. Again, you're looking for the blue words to try to label things. After about 24 hours, what's going to be happening is what's going to be a zygote after this point is going to start the process of traveling. Well, it actually really already started that process, but it's going to continue the process of traveling down the fallopian tube. About a day later, it starts to divide and we become the 2-cell stage, the 4-cell stage, the 8-cell stage, the 16-cell stage, so on and so forth. Right around here, we become that solid ball of cells, the morula. We keep traveling. Eventually, we become the hollow ball of cells, the blastula. And this is what's actually going to implant. And again, we should be implanting in the upper portion of the uterus, someplace up there. If we get to be too low, that's not okay. We talked about ectopic stuff. And if we implant up here, tubal pregnancy, that's a type of ectopic. That's not okay either. This picture does also give you trophoblast inner cell mass labels, and so it gives you that sort of anatomy as well. If you really wanted to study well and overachieve, you could even come onto this picture and label primary follicle, secondary follicle, mature follicle, corpus luteum, corpus albicans, because all that stuff is off over there too. Okay. Explain the process of implementation. So this is the picture from your book that's trying to show you what's happening. What's going to happen is the trophoblast and the endometrium are going to start to fuse together and essentially sort of melt each other, and you get a lot of wild dysplasia that happens where the cells become very irregularly shaped. It becomes very difficult, nearly impossible on microscope examination to be able to differentiate mom cells from baby cells at this point. Genetically you'd be able to, but we're not going to play around with that game. But what's happening is mom and baby are literally fusing together. Mom's immune system is unhappy about this, but at the same time, it's got to happen. And so there's chaos, but it's controlled chaos, essentially, is what's happening during implantation. There are women that are considered to have, quote unquote, a hostile uterus, and that's when her immune system doesn't control the chaos very well. And the uterus essentially rejects this like it's foreign tissue, which technically it is, but the immune system's not supposed to be that hyperactive within the uterus. What structure secretes the human chorionic gonadotropin? So the trophoblast from the baby is what actually secretes this. HCG is the hormone that actually gets detected during a pregnancy test. So when you pee on a stick and it tells you you're positive or negative, it's really telling you whether or not you're making HCG, because the only natural time that hormone is found in a human body is when a baby is secreting it because you have made a baby. The weird side note I always like to explain here, that is true. It sounds crazy, but it is true. If a guy pees on a pregnancy test and it pops positive on that test, that means his testes are secreting HCG, and that's a really strong indicator for testicular cancer, and so it would mean he should go get that checked out by a doctor. This shows you the hormones that happen during a pregnancy, and notice it's the early stages of pregnancy where you get this huge spike in HCG. As it starts to taper off, estrogen and progesterone really start to take over and maintain that pregnancy through to the end. The placenta. So it is a dual organ. It is made by both the baby and the mom. However, the two tissue types, once we get it organized, mom stuff and baby stuff is really close to each other side by side, but it's important that you understand that mom's blood and baby's blood do not directly mix with each other. While there are bad things that can cross the placenta, like hepatitis B, for example, which is, we talked about that in the digestive system, the placenta, because it's two separate tissues, only stuff that can actually diffuse through plasma membranes can pass between the mom and the baby. And so it's trying to make sure only the good stuff passes. Sometimes, however, it does allow some things that shouldn't be getting through to get through. But no direct exchange, no direct mixing, let me say it that way. This is a placenta. We'll talk again about this a little bit later on. One of the things people don't realize is after you give birth to a baby, you have to also give birth to a placenta. It used to be called afterbirth. I'm sure some people still call it that. Okay, so we're a little bit later in development. You can tell that this is an embryo. You can start to see they've color-coded different tissues out here. This area is the chorion. So again, the trophoblast becomes the chorion. The chorion has all of these little villi or extensions that the baby makes. I like to think of them like fingers. The baby is reaching these creepy placental fingers into mom and grabbing onto mom, so that one, we don't dislodge, and two, we're exchanging materials really easily between mom and baby. So the chorionic villi are what are going to bring the baby's blood supply near mom's blood supply. Placenta should be fully functional by 3 months. Prior to that, the corpus luteum was what was secreting all of the hormones. This is just to show you, there are a lot of women who you can't tell are pregnant when they're 3 months along, and so she has missed a couple of periods by this point. She should be aware that there's something going on, and yet a lot of people around her maybe don't know. After you make it through 3 months, that's the first trimester, and that's usually when people start telling others that they're pregnant, and that's because that first trimester is fairly risky. There's really good high probability that things don't progress like you think they're going to. And if you've told people that you're pregnant and then you lose the baby, well, now you have to tell them afterwards you didn't. So that's why a lot of people wait until after that first trimester to start telling them. Chance of success is better after that point. Oh, that was where I wanted to stop this video. We'll continue on. No, it wasn't. Never mind. I'm going to keep going. I forgot where I wanted to stop. So which hormones are the placenta going to produce? HCG early on, but again, estrogen and progesterone later. I showed you that graph for what it means, or where you get your different spikes in different hormones earlier. Okay, so next, we were at the blastula stage. What happens next is we start to get this infolding or pouch that develops within the blastula. This is going to be a primitive gut. That opening, because we're deuterostomes, the second opening is what becomes the mouth, so eventually this opening is going to end up becoming our anus, and so that kind of gives you an orientation as to what's going on here. But in the process of folding that pouch, again, some of our cells start down a differential path, where some of them are going to become ectoderm, some of them are going to become endoderm, and some of them are going to become mesoderm, which you can't see in this picture, but that's another layer that we form in human beings. Extraembryonic membranes are membranes that the baby makes, but they're not actually part of the baby. They're layers that are outside of the baby that help with the development of the baby while the baby is in utero still. There's going to be 4, and one of them we've already talked about, the chorion. So the other 3 that we need to talk about are the amnion, the allantois, and then the yolk sac. And so that's what we're going to hit on a little bit next. So, amnion. This is an ultrasound of a pregnant woman. You can see the baby labeled right here. You can also see there's a sac around the baby. That sac is the amnion, and it is filled with fluid called amniotic fluid. When a woman's water breaks, it's actually the amnion that breaks, and then the water that comes out is actually the amniotic fluid. And so what this is doing is creating a nice, watery, soft, happy environment for the baby to grow up in, essentially. Now this is a more primitive picture than what this one is. The baby would be inside of this amniotic cavity. They did not draw that in this picture, but it would be inside of that blue spot. Then if my video feels like playing, I'll try to make it play. I don't know why it works sometimes and it doesn't work other times. It's always in a mood. So I'll just kind of explain since my video doesn't feel like working. We're doing a C-section here in this. This little yellow sac around the outside, that's the amnion, and it is still intact in this woman and over this baby, and you can see the baby's face going on right there. I don't know why my video doesn't want to play for you guys, but you could actually see the baby moving around in it if the video were going to play, and then it tears right down there, and you can see all the fluid gushing out, and that would be the amniotic fluid. Just as an aside, babies don't usually get born with an intact amnion. It used to be considered good luck if your baby was born this way, so this was an unusual birth. Okay, so this is the fluid that's outside of the baby, and a little side note that I'll give you, when they do an amniocentesis, they use a needle to collect some of that fluid because there are some baby cells in that fluid. We can test those cells for different genetic abnormalities that might run in your family, or if you are a late-stage mom, a geriatric pregnancy, they can do chromosomal tests to see if they might have Down syndrome or Turner syndrome or something like that. The yolk sac. So it's this yellow area going on off over here. It does different things in different kinds of organisms. Like in a chicken, it does actually become the yolk. But if you've ever raised chickens, like in elementary school or something, one of the things that you'd notice is that's the area where the first blood vessels actually start to form, and it makes our first blood cells. It does that for human beings as well. It just doesn't stay as obvious as a chicken yolk afterwards. The allantois is what's going to help form the umbilical cord that attaches the baby to the placenta. So here's just a picture of a nice, healthy, happy umbilical cord. They do usually well, if there is a dad in the room, they often will ask if dad wants to cut it so dad can feel like he's participating in the whole thing. I don't know, he's just been staying in there for the last 12 hours. You might as well ask him to do some minor surgery here at the very end, right? Alright, so here's where I'm giving you a picture so that you can label all of those extra embryonic membranes that we just went over. So again, that blue layer is the amnion, you got your yolk sac, you have your allantois, and then your chorion is what helps form the placenta out here. So this is, like it says, roughly a 5-week-old embryo. We've got arm buds, leg buds. We do still have the tail. The eye is starting to form. The brain is doing little brainy things inside of there. These little They're gill slits at this point. Some of the gill slit is going to end up becoming portions of the ear, and some of it's going to end up becoming the mandible. So a lot of this is going to migrate and move to other places. By this stage, we're starting the formation of the heart. The heart's not beating at this stage, but it's usually starting to form right in that area. So that's what a baby looks like when they're about 5 weeks along. Alright, and then this was the slide I was going to finish on for today. It goes back to those germ layers that we talked about in the first half, and I already said all of this, so you can just pause it to get it. We will continue on with gastrulation, which I define now, even though we already defined what a gastrula was. So we'll continue here next time.